agriculture, engineering and science activities in the region. The college comprises 5 schools: agricultural, earth and environmental sciences, engineering, chemistry and physics, life sciences and mathematics, statistics and computer science. Engineering is offered on the Howard College campus, agriculture on the Pietermaritzburg, and all the other sciences on both the Pietermaritzburg and Westville campuses. Exceptions are agricultural engineering, which is offered in Pietermaritzburg, and geology, which is offered at Westville only. To compete with the best and to meet the challenges of the 21st century, South Africa needs highly skilled professionals in the areas of agriculture, engineering and science. We offer a wide array of interesting and innovative degree programs for different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree and one of only 2 universities in the country to offer a BSc in land surveying. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science, and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural disciplines for a 4-year Bachelor of Science in Agriculture degree. These are more career-orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the envy of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agricultural engineering. Adjoining Ukulinga is the Bisley Valley Nature Reserve, which supports a diversity of plant and bird species, as well as some large mammals. It provides a rich learning and research environment for staff and students. Based on the Westville campus, the college possesses unique facilities for research into higher voltage power transmission. These facilities are available in only 3 other places in the world and form part of the ESCOM-sponsored Science and Technology Innovation Park. The Science and Technology Education Center showcases the college's scientific achievements and promotes the public understanding of science, engineering, and technology within the region. The college houses a wide range of sophisticated equipment in its microscopy and microanalysis units, staffed by highly qualified and skilled technical specialists. The College of Agriculture, Engineering and Science prides itself on its graduates who are highly sought after. In a market characterized by a shortage of qualified professionals in science and technology, our graduates are readily employed around the world in the fields of agriculture, science, commerce, industry, engineering and research, to name just a few. Research plays a major role within the college and operates at the cutting edge of technology. The college has the highest research output in the university and produces the most research publications. There are over 60 well-established research groups and centers within the college. The African Center for Crop Improvement caters for students from all over Africa. It is one of only 2 African plant breeding programs on the continent. Based in the School of Agricultural, Earth and Environmental Sciences, the African Center for Food Security focuses on alleviating hunger in Africa. It offers food security training programs, undertakes academic and applied research, and provides policy and program support. Promoting sustainable engineering practice is the Center for Research in Environmental, Coastal and Hydrological Engineering, or CRESH. It deals with the interaction between engineering and the natural environment. The Mechatronics and Robotics Research Group in the School of Engineering provides an excellent research and education environment for its senior undergraduate and postgraduate students. Judged one of the best research centers at the university, the Center for Plant Growth and Development focuses on high-quality research in the plant-based disciplines of plant physiology, plant molecular biology, and ethnobotany. The Center for Quantum Technology deals with physical phenomena at the microscopic level. Quantum physics led to the development of the transistor and the laser and is currently being used to develop quantum computers and quantum cryptography. The college
Good day, students. Thank you for joining us this morning. It's wonderful to have so many of you join our revision Zoom session this morning. My name is Mrs. Jerusha Naidu, and I'll be doing some biology revision with you this morning in preparation for your paper 1, which you will write early next week. Okay. I'm just going to share my screen and we're going to jump straight into it. Okay, can everybody see that? You can just give me a thumbs up. I'd like this session to be interactive, so you're more than welcome to unmute yourselves and shout out an answer if you need to, or ask a question. It's we are open to all types of interaction this morning. And if you are also not understanding something, you're more than welcome to ask me. Okay, can I get a thumbs up just to make sure we're all on the same page and that you can see my screen, which should say Cassie Matric Helpline. Great, okay. Okay, so this morning we're going to just look at two of the major sections that you will be covering in your paper one, okay? And that is the biggest section of your paper one is human responses to the environment. And that's going to be worth 54 marks of your paper, which is about 1/3 of your paper. So it's very important that you know this section well. So I'm going to give you, we're going to go through lots of past papers, which I'm sure you may have come across in school, but maybe your teachers didn't go through it in detail with you, or you just attempted it on your own. So we're going to definitely go through these questions from past papers based on human responses to the environment and the endocrine system and homeostasis in humans, okay? And the latter section is worth 34 marks, okay? So those are two major components of your paper. Okay, the other sections that will be covered will be, in your paper one will be covered in my next session with you on Thursday. So please do join in for those sections. For example, we will be covering reproduction in humans and vertebrates and plants' responses to the environment. Okay, so those smaller sections or the second half of your paper one will be covering in our next Zoom session on Thursday. Okay, so we're just going to do these two major sections today. So I'm going to be going through some questions with you and highlighting some key concepts that you will need to know. And if you have any questions along the way, please feel free to pick up your hand and ask me, or you can even put it into the chat. There are staff members that are here to assist you and also convey the question to me. So I'll be more than happy to answer them for you. Okay. Okay, so in the human responses to the environment section, that is, as I said, your biggest section. And that comprises of the nervous system, which is the central and peripheral nervous system. Okay, and then you also have to learn about the sense organs, and that would be your ear and eye. Okay, and for the endocrine system and homeostasis in humans, you'll need to know about hormones and negative feedback systems in homeostasis, as well as the organ of the skin. Okay, so we'll be talking a bit, I'll be just revising some of the key concepts with you as we go along, should it come up in these questions. Okay. Okay, so let's jump straight into it. If we have some time at the end, we can do, I'm doing these short answer questions with you first, because that's the bulk of your paper. If you've done these past papers before, you'll notice that there is a section C in your past papers, but you are very lucky this year. You're not having any essay component in your matric paper. So I suggest you use that to your advantage. There's much more short answer questions, so you can really score your marks. Okay, you don't have that 20-mark essay question that was present in many previous year exam papers. Okay, so your section A will be 50 marks, and that will be your like one-word answers, name the biological term, multiple choice, matching, that type of thing. And section B will be these type of short answer questions, okay, which will actually work out to about 100 marks. Okay, so we're going to focus on these short answer questions. Okay, some of them are also one-word answers and things, and some of them are a bit longer, like for 4 to 6 marks. Okay. Okay, so we'll start off with the human responses to the environment. Now, as we said, the nervous system comprises of the central and peripheral nervous system. So can anybody tell me quickly what structures are involved in the central nervous system? You can just unmute and shout it out, or you can put it in the chat. Any takers? And please feel free and relaxed in this session. All your fellow students are here and everybody's in the same boat. So don't feel shy.
It's the brain and the spinal cord.
Very good. That is correct. And what's in the peripheral nervous system?
Cranial nerves and spinal nerves.
Very good. Okay. So what do you need to know about those two things? See, as we go along, you can jot down a summary. If you don't have the study guide, you can just make your own sort of summaries where you don't have to now go through, sift through a whole lot of information from your textbook, okay? If you don't have the study guide, you're just going to make these quick notes. Central nervous system dash brain spinal cord. So it's easier for you to quickly revise these things the night before your exam, okay? It's also important for you to know what components are in each section as well. In general, in this paper, in previous papers as well, you will see that sometimes they integrate different sections. And I've got an example of a question later on in this PowerPoint, which I'll show you, which came from 2020's past paper, okay? Where your nervous system is integrated with the eye, for example. Okay, so you are going to find that there's integration of different sections. Okay, so don't freak out when you see that in the paper or get confused. Okay, so that's why I'm saying try and just make a little summary of what's in what section, if that makes sense. Okay, so let's start with the first one. I'm just going to type the answers for you as we go, but I'd first like you guys to try it, okay? So, and we'll go through these ones quickly. We're starting off easy with this nice, easy picture here. Okay, so obviously you can see a brain in this first diagram, okay? And it's showing you a part of the human central nervous system. Okay, so the brain is part of it and the spinal cord, you can see a small segment of it there. Okay, so identify part A. What's A? Very easy.
The spinal cord.
Correct. Thank you. And what is C? The thing that holds the two hemispheres of the brain together.
The corpus callosum.
Very good. Okay. So now, can you just tell me, we will use this to revise. What else does the spinal cord in A do? What's the function of the spinal cord? Seems like a very easy question. It's going to transmit nerve impulses to the brain. Okay, very simple. Okay, so we've identified part A and part C. Now write down the letter and name. Okay, so this is for 2 marks, so don't mistakenly leave one of the other out. Okay, so the letter and the name of the part that has the center for interpreting taste. So where is taste interpreted? Just from your memory from learning now. Where is taste interpreted?
The cerebrum.
Very good. Now on the diagram, where is the cerebrum?
I think it's D.
Yes, that is absolutely correct. You've scored yourself 2 marks. Okay, next one. Which part regulates the heart rate? From what you've learned?
Hypothalamus.
The
hypothalamus.
Okay, but you can't really see that on this diagram now. So from what you can see on the diagram, which part will regulate heart rate? Any other takers?
Isn't it the medulla oblongata? B.
Very good. The medulla oblongata, excuse me, will regulate heart rate and diameter of your blood vessels. Okay, so which label is the medulla oblongata?
B.
Sorry?
B.
No. Yes, actually, it is B. Okay. And then which part is responsible for motor coordination?
A.
The spinal cord? Any other takers?
I think it's A.
Okay, anybody else got something different? So which part of the brain is responsible for motor coordination?
E.
And what is that?
The cerebellum.
Very good. Okay, so the cerebellum is involved in coordination of voluntary movements such as running, walking, posture, and balance. Okay, and that is the label E. Okay. Did anybody miss out those? Sorry, I forgot to type it as I'm going. Anybody need me to repeat any of those answers? You can just raise your hand. If not, I'll move on. Okay, so we're okay with our basic brain at this point. Let's try another example or a question. Okay, so I'll remember to type your answer in here for you now. Okay, so what do we have here? What are those funny looking alien things?
Neurons.
Very good. Now, you can see 4 different types of neurons there. Okay, they labeled 1 to 4. As soon as you get a question like this, your brain must be using those neurons, okay? And they should be firing rapidly. Tell me, what is the neuron number 1?
Sensory neuron.
And how do you know that, young man?
It doesn't have a cell body.
Well, it does, but it's a very small one.
A small cell body.
Here's it here. Can you see that? I don't know if you can see my cursor, but there's the little cell body, but it's the position of the cell body that
It's in the middle.
Will tell us. It's in the
middle.
Very good. Okay, so we can see that our small cell body is in the middle of our neuron or our axon. So we know that is a sensory neuron. And what does a sensory neuron do? What's the function?
Conducts nerve impulses from receptors to the central nervous system.
Very good. Okay, so it's gonna conduct a nerve impulse from a receptor to the spinal cord in the brain. Okay, and now that looks quite different to, let's say, a neuron number 3, which is what?
A motor neuron
Very
with a myelin
good.
sheath.
Yes, correct. And what is the function of this motor neuron?
Transmits impulses from the spinal cord to the effector.
Yes, that is correct. Okay, so can you see the difference in function between the sensory neuron and a motor neuron?
Impulses from the central nervous system to the effectors.
That is correct. Okay, so a sensory neuron is going to pick up that sense that it's getting from a receptor, and a motor neuron is going to make you respond to that sense. Okay, so the sensory neuron is the receptor, and the motor neuron is more of an effector. It causes an effect. Okay, so there's another type of neuron actually that's visible in this picture, and those are called interneurons. Okay, and those are numbers 2 and 4. Okay, and interneurons are obviously, as the name says, going to be in between your sensory neuron and your motor neuron. Okay, and I'll show you an example of it in another question, in the next question, where you're going to find these interneurons in your spinal cord and even in your brain. Okay, so let's get answering this question. Which, it says, give only the numbers, which is 1, 2, 3, or 4, of 2 neurons. Okay, so it's going to be 1 mark each. 2 neurons that transport impulses from the receptor to the central nervous system. So we just said one of those.
1 and 4.
1 and 4.
Yes, okay, that is correct. We'll have faster transmission of impulses. Think about this one.
1 and 3.
What's it?
1 and 3.
That is correct. And why?
Because 1 and 3 has a myelin sheath.
Very good. And what is the purpose of a myelin sheath?
It covers the axon that's hitting the axon, which makes impulses go fast.
Correct. Okay, so the axon is actually insulated. You know, the same way as your normal cords around your house are insulated.
Yeah.
Or if a cord breaks and you use that black insulation tape and then it works again. Okay, so that it allows the transmission of that impulse. Okay. However, you can see that there's these little tiny breaks in this myelin sheath. Those are called the nodes of Ranvier. So the impulse actually jumps to each node. So can you see that this impulse is not going to travel all the way along this myelin, all the way along this axon. It's actually going to be as fast as like a rabbit jumping. So it's covering more of a distance in a shorter amount of time. So that obviously you want your impulses to be transmitted very quickly. Okay, so the whole point of an insulator is to transmit that signal and to do it quickly. Okay, so that your signal can be transmitted fast. So the signal will actually jump to each of these little gaps in the myelin sheath. Okay, so it's actually jumping down this axon. Okay, and that actually that is what will speed up the transmission of that impulse. Is everybody with me? Do you have any questions about that? Okay, if none, we'll move on. Okay, so which 2 neurons are damaged if a person can feel the stimulus but is unable to react? And a quick note, this type of a question has actually come up in 3 past year papers. So know this answer, just in case it comes up this year. Which 2 neurons are damaged if a person can feel the stimulus but is unable to react? Which means that your sensory neurons are fine, but your motor neurons can't react to that stimulus. So what number is that going to be? Sorry?
2 and 3. 2 and 3.
2 and 3. Okay. Any other options? Yes. Okay, well, it is 2 and 3. That is correct, because an interneuron is, as we said, between the sensory neuron and the motor neuron. So once the sensory neuron picks up that signal, it can't. There has to, if somebody cannot react to that signal, maybe there's a problem with the interneuron that's having a problem transmitting the signal to the motor neuron, or the actual motor neuron cannot cause an effect for that person to be able to react. Okay, so the interneuron and the motor neuron could be damaged if this person cannot react to a stimulus. Okay, so that is that question that came from last year's end of year exam paper. Okay, let's move on. Is everybody okay so far with just this first part that we've been doing? Any questions?
No.
All right. Okay, so now we're going on to a bit more of a hectic question, as you can see. This was from 2018's paper. Okay, so as we said, and I don't know if you can see this properly. Okay, so if you look here, I'm just going to zoom it for you. Okay, because it was a bit difficult to see. So can you see the difference between this side of the diagram, pathway B and pathway A? Okay, so pathway A is going, follow my cursor, it's going into the, what is this, by the way?
I think it's the spinal cord.
It is, okay, and it is, as you can see, this now this impulse is going through from this sensory organ through the spinal cord, and it's just turning around there and then going back into an effector muscle. Okay, compare that to pathway B, getting a signal, a sense. It gets to the spinal cord and then it goes to the brain, and then it comes back through the spinal cord and then goes to the muscle. Okay, so that's the difference between pathway A and pathway B. Now, the question is, which pathway A or B reflects a reflex arc, represents a reflex arc?
A.
And why would you say A, young lady?
The pathway and the neurons are passing through the spinal cord only.
Very good. Okay, so what is a reflex arc?
It's a pathway taken by an impulse in response to a stimulus.
Right, but that could be any stimulus. Why would this impulse need to take the route of a reflex arc? What's the importance of a reflex arc? You could get a question like that, for example. Maybe they could ask you the definition of a reflex arc. Well, actually, it's right there in question 2.3, 3.2.3. The importance of a reflex arc. So what's happening in pathway A is that the stimulus is being received, it's going through the spinal cord, and immediately, almost involuntarily, there is an effect being elicited from that signal that was received from the sense organ. Okay, it's only going through the spinal cord. And why is it only doing that? Because the body is in danger, maybe, or a reflex action needs to occur, which means there is no time for the signal now to go to the brain, and then the brain sends a signal back, and tells the muscle what to do. There's no time for that. You could be dead in that time. So a reflex arc is an evolutionary phenomenon where the stimulus just only passes through the spinal cord such that an effect can be elicited extremely quickly. Okay, so it's a very rapid response in a rapid response to prevent danger to the body. Okay, and there's no, obviously you don't think about it. If you've ever been to the doctor, the doctor may tap your knee and your knee just flicks out without you actually thinking about anything. That's an example of a reflex. Okay, it's an involuntary reaction to a stimulus. Okay, and it's very important that people have reflexes in order to prevent damage to the body extremely quickly. So that is why the stimulus is going from the sense organ only through the spinal cord and into the muscle to cause an effect very fast. Okay, is everybody with me? You okay there? Yes or no?
Yes.
Okay, so we said a reflex, it's a reflex arc because it's only going through the spinal cord, right? Okay, next question, and we've just discussed 2.3. Identify the part of the nervous system represented by 1, which is the whole diagram. There's the number 1 there. So what part of the nervous system is this? It is obviously the central nervous system. Okay, that's an easy 1 mark. Okay. Explain one way in which the myelin sheath is important in the functioning of neurons. And we just discussed this in the previous question. So one way in which the myelin sheath is important in the functioning of neurons. Okay, so it's asking you for a function of that myelin sheath. So when questions ask for importance, it means you must say the function or the relevance of that. And in turn, if you are asked for the function, that will be why it is important, if you understand what I'm saying. Okay, so just look at this type of phrasing because it's something that will definitely come up in your paper. Okay, so explain one way in which the myelin sheath is important in the functioning of neurons. It insulates that axon so that impulses can be transmitted very quickly. Okay, and that is just 2 marks. Okay. Then the next question, describe how the person would be affected if the axon of neuron 2 was cut. Okay, so if you cut this axon of the neuron number 2, what neuron would that be? That would be your motor neuron because it's going into the muscle. Okay, so if you had to cut that neuron 2, it means that the person, again, as I told you, this question seems to come up a lot. It's for 2 marks. The person, it means that the person can receive the signal, but the person cannot react to the signal. You with me? Because this sensory neuron is fine. It's going into the spinal cord. It's receiving that sense. So the person is aware of the sense, but there cannot be a response to that sense because this motor neuron has been cut. Are you with me? So know that type of question, okay? Because as you can see, it's come up in 2018 again. Okay. Okay, and then for 6 marks, a big question. Describe pathway B. for 6 marks. And I've just put the answer here for you just to save time. Okay, the types of things that you would need to include in your answer. Okay, so pathway B is the normal pathway that any sense is going to take on. Okay, so the receptor receives the stimulus from, let's say, your finger. Okay, and now that stimulus is converted into an impulse. Okay, and that is obviously going to be transmitted by the cell body, which is located right here in this little region. What is this region called? Anybody knows? This little bump here. That is called a?
Synapse.
No, it's called the dorsal root ganglion. And that is where the cell body of the sensory neuron is housed in the spinal cord. Okay, the synapse, young man, is what? What is the definition of a synapse? It is the connection between 2 neurons. Okay, it's a gap between the end of an axon of one neuron and the dendrite of another neuron. So a synapse might occur somewhere over here. Can you see between the sensory neuron and this interneuron here? So a synapse would be at this point. Can you see that? The synapse would be more in this point here. This little bump here is the dorsal root ganglion. Okay. So then the receptor, as we said, receives the stimulus and converts it to an impulse. And then that impulse is then transported by the sensory neuron, by the spinal cord and an interneuron to the brain, and particularly the cerebrum of the brain, according to this diagram. The brain or cerebrum will then interpret that signal, okay, that impulse. So you must use these key words like stimulus, impulse, signal, okay? And once it's interpreted that signal, then obviously your body knows what to do with it. So a message will be sent from the cerebrum. An impulse will be sent from the cerebrum down through the spinal cord once again. And it will now be transmitted through a motor neuron, okay? And the motor end plate is now located, the motor end plate of that neuron is now located in a muscle, for example. So that those dendrites of that motor neuron will receive that signal from the brain and elicit a response in the muscle via the motor end plate. Okay, so that will bring about a response. Okay, so in this question, I've taken this from the memo as well, so just to show you. Okay, in this question, you can see that they've said that this point here is compulsory. Let me just highlight that for you. Where's my highlighter one? This point is compulsory, and this point is compulsory, which means you have to get those two, you have to say these two things in order for you to get the minimum of 2 marks there. And then you can write any of the other 4 points. Okay, so you need to be able to say that a sensory neuron transmitted the signal to the brain, and then the brain interprets the signal. So the brain is key in this normal pathway, okay? A non-reflex arc pathway, okay? And then you'll get a mark for any of these other 4 points of explanation. Okay, is everybody with me there? Are you fine with answering a question like this for 6 marks?
Yes.
Okay, very good. Okay, so let's move on. Now, as I was saying before, here's an example of where they are putting different sections all in one question. You've got brain. What is this? What's this part of and this part of? They are part of the ear. Okay, so you've got two different sections here being mixed up. So that's why I chose this to go through. Okay, this was from last year's paper actually. Okay, so identify part labeled A.
It's Medulla Oblongata.
Very good. So as you can see, they keep asking you that. So they will also definitely ask you for labels of the brain at some point in your paper. So you must be able to label the brain, the ear, the eye, and well, just for these two sections now, at least, and even the spinal cord maybe. Okay, you must know labels. I'll give you some tips at the end as well, just that pertain to these sections, but also the rest of your paper one. Okay, so back to the question. A is the medulla oblongata. What's B? Sorry, let me type it for you.
Cerebrum.
Correct. What's H? Starts with the E.
The station tube.
Very good. Okay, and then next question is, and these would all be for 1 mark. Okay, give the letter and name of the part of the ear that absorbs excess pressure waves from the inner ear. And again, this question always seems to come up. So you need to know this answer. What little part absorbs excess pressure waves from the inner ear? Give the letter and the name.
G. The round window.
Very good. So the round window is involved in absorbing those pressure waves from the inner ear, which because it prevents reflection of those pressure waves into the middle ear. Okay. Okay. Then name the receptors found at E. Where's E? What organ is that? I mean, what part of the ear is E? What's that label?
The cochlea.
Okay.
The cochlea.
Very good. So, but the question is asking now, in the cochlea, name the receptors. So you can't just say cochlea, you have to say those organs of Corti are the actual receptors in the cochlea. Okay. Everybody with me? Make sure you're always answering the actual question. Okay. Then explain why damage to part A can lead to instant death. If you damage it, remember we said that A is the medulla oblongata. Let me recap what's the function of the medulla oblongata. Again, what is the importance of the medulla oblongata?
Wash your face and have breakfast.
Function of the medulla oblongata?
Regulates heartbeats.
Yes. Heartbeat, breathing, involuntary muscle movements, coordination of swallowing, coughing, sneezing. But why? So now we know the function of the medulla oblongata. That's its importance. So now the question is asking you, if you damage that, why would that lead to instant death? So if you damage a part of the brain that's involved in breathing and heartbeat, obviously you're going to die. You know,
Yeah.
you're not going to be, your heartbeat's not going to be regulated. Your breathing is not going to be regulated. So it's going to lead to death. Okay, so that's a simple answer, but the way this question is phrased is not straightforward. Okay, and they like to use this type of phrasing about damaging a part. As we saw in the previous question, they asked you if that neuron was damaged, which means that you need to know the function of that part and the importance of it. So then you'll know if it is damaged, what will happen. Okay, so function is absolutely, absolutely important. You must know the function of these parts when it comes to particularly these organs that we're talking about, the brain, the ear, the eye, the skin, all of those types of, all these organs, you must know the functions of these labels. Okay. Okay, so describe how part C responds to impulses received from part D. Okay, so part C is what? The
cerebellum.
Cerebellum. And part D is, what is this? Those are your membranous semicircular canals. Okay, so let's just talk about what the semicircular canals do. They are actually filled with fluid, okay? And when your head moves, that fluid also moves. And then that will now transmit a signal which will be interpreted by the cerebellum. Okay, so the cerebellum will interpret this head movements. Okay. Because the cerebellum is involved in balance, in maintaining balance. Okay, so for 3 marks, this question needed to, the answer needed to be that the impulses will be interpreted and sent to your skeletal muscles to maintain balance. So that is how part C will respond to impulses from part D. Okay, so these semicircular canals, as your head moves, the liquid moves, and that will transmit a signal to the brain, particularly the cerebellum. And the cerebellum will now send a signal via the spinal cord to the muscles for you to maintain balance. Okay, so in this case, you needed to know that the semicircular canals and the cerebellum are involved in maintaining balance and are involved when you move your head. Okay, everybody with me? Are we still okay?
Yes, ma'am.
Yes.
Very good. Okay, so in older people, part F of the ear may harden. What is part F? Then explain how this condition may lead to hearing loss. Okay, so remember now, in the ear, can you just see how this question is so integrated? You have to jump from section to section in your brain, okay? So it's a window of some sort. So in your ear, you've only got the oval window and the round window. Okay, so that bigger window is your oval window, and the round window is, as we said, was G, okay? So F is the oval window. So what is the function of this oval window? It actually transmits vibrations from those 3 bones, as you can see here. These are called ossicles. From these ossicles, it will transmit that signal into the inner ear. Okay, so that's the function of that oval window. So the question is asking in older people, if this oval window has to harden, so it cannot perform its function of transmitting that signal, that is how it's going to lead to hearing loss. Okay, so this oval window will not be able to vibrate because that is the way that these ossicles transmit the signal of sound. Okay, it vibrates and transmits that vibration via the oval window into the inner ear. Okay, so if this oval window cannot, if it's hard, it can't vibrate. Okay, so there's fewer or no vibrations that will be carried into the inner ear. Okay, and or fewer or no pressure waves will form in this cochlea, because this is a diagram of the inner ear. Okay, so here's this oval window here. Okay, if you can see that. And that signal gets transmitted into the cochlea there. Okay, so if there's no signal that's transmitted into the inner ear, there'll be no stimulus or stimulation of the organs of corti, which are in the cochlea. Okay, those hair cells. And therefore, there'll be no transmission of an impulse via this auditory nerve to the brain. Okay, so if there's no signal from the auditory nerve to the brain, it means that the brain has not received any indication of a sound, which is hearing loss. You cannot hear. Okay, so that is a question that was out of 4 marks, but can you see that you have to explain each and every step of this pathway of hearing? And yet all they've asked you about was the oval window. Are you with me? So you need to know, I would say, pathways, okay, of transmission of an impulse, of sound, of hormones, that type of things. Okay, so pathways. Okay, then describe accommodation of the eye or distant vision. Okay, that just randomly popped up in this question. It's not referring to this diagram, but it's still part of the question, 2.2. And this is extremely important. This question, you must know it because it's always going to come up. Okay, the eye, there's only a certain amount of things that they can ask about the eye. Okay, and this is now our first time going through the eye in this practice session. Okay, so accommodation of the eye, which means accommodation means that the eye is adjusting to see things at different distances. Okay, so let's just talk about accommodation. Okay, so describe the accommodation of the eye for distant vision. Okay, so you're trying to see something that is far away. Okay, so distant vision, which means something that is beyond 6 meters away from you. Okay, and that is for 5 marks. Okay, they're definitely going to ask you a question about accommodation or light stimulation and how the eye changes. Okay, so please, you must know that. That's definitely going to be a question about from the eye. They can also ask you to label the eye, and then in one of the sub questions, there'll be, how is your eye going to change to be able to see distant objects? Okay, so then you'll need to say things like this. The ciliary muscles are actually relaxed, okay? And sorry, I should have put a picture of the eye there so I can show it to you. But these ciliary muscles now, they are involved in actually controlling the shape of the lens, okay? So they can adjust the shape of the lens depending on whether you are looking at something near or far away. For example, on your cell phone, when you're taking a picture, okay, your phone will tell you, move your phone further away from the object. Why? Because that lens needs to focus. You with me? So in the same way, your eye has to adjust such that it can see things at different distances away. And to adjust that lens in your eye, the same way that your camera lens on your phone adjusts, the thing that adjusts your lenses are your ciliary muscles, okay? So this, in order to view something that is far away, that ciliary muscle must be relaxed, okay? And then the other parts of the eye that's attached or is a consequence of the ciliary muscle is the ciliary body and the choroid layer, okay? So the ciliary body and the choroid layer then moves backward or away from the lens, okay? And the suspensory ligaments are tight, okay? Which means that it's pulling on this lens, okay? And that creates tension. And that means that the lens is now being made more taut, which means more tight or like firm, okay? So the ciliary muscles actually are going to pull this lens to be more tight. And that makes the lens less concave, okay? So if something is tight, it means it's more flat, okay? It's more straight. For example, if you have like any cord, your charger cable, and you pull on either ends of the charger cable, it's going to become straight. If you release or if you relax your hands, your charger cable will be more curved, okay? In the same way, the ciliary muscles will pull that lens so that the lens is less convex, which means less rounded, and it becomes flatter and straighter. And that now causes the light rays from that distant object to be refracted less, which means it's bent. Those light rays are bent to a lesser degree, okay? And now those light rays can actually fall on the retina, which is that back part of your eye, okay? So the retina will actually receive that image clearly because those light rays are actually landing on the retina and not past the retina, okay? So that is what your eye has to keep doing to adjust to any object. It has to keep focusing. So your lens is going to always be changing to be less or more convex, such that objects that you can see, that you want to see far away or close by, the light that's coming from those objects can be refracted and land. Those refracted rays must land on your retina. That is the only time you're going to see something clearly. Okay, so your 5 marks are coming from saying that the ciliary muscles are, what are the ciliary muscles doing? Okay, and the other things that are involved with the ciliary muscles, like the choroid body and the suspensory ligaments. And then the ciliary muscle is having an effect on the tension of the lens. Okay, so that's the second thing that you need to make sure you mention. And then that lens is now, the purpose of the lens is to refract the light rays, and that light rays must now fall on the retina. Okay, so those points are key. That is the sort of framework for you to answer any of these accommodation of the eye questions. Okay, whether it's near an object that's near or distant. Okay, so please learn this. Okay, it's definitely going to come out somewhere. Is everybody understanding me? Is everybody fine with this?
Ma'am?
Yes.
Can you please emphasize more on the retina and the yellow spot? Is it the same thing?
Okay, so the retina is a region of the eyeball. It is the whole back of the eyeball, right? So if you take your palm and you cup your palm, okay, that your whole hand will be your retina, okay? but the yellow spot is just one place on that retina. And that is where there's only cones. Okay, so you know you have your cones and your rods. Your rods see black and white and your cones see color. So in the yellow spot, there's only cones, which means that at that point, that is where you can see very clearly. That is your spot of most clear vision. Okay, so the retina is a big region. It's that whole back of your eye. But the yellow spot is just one place on that retina that has only cones, and that is where you can see the most clearly. Okay, so an image can form on the retina, but which means you can see it clearly. But if that image falls on the yellow spot, then you can see it the best. You can see it the most clearly. You and me. Is that answering your question? Do you understand that distinction?
Okay, ma'am. Thank you.
Okay. Any other question on that? Okay, I'm going to move on. Okay, so we're back to the ear. Okay, this was from 2018's paper. Give one function of part A. Sorry about the birds in the background. What's part A? Anybody?
That's the auditory canal, and it's the tube leading to the eardrum.
Okay, that is correct, the auditory canal. And what is part E?
Eustachian tube.
Very good. What is part F?
The round window.
Very good. So as you can see, this is coming up again. Okay, so you must know these things. Sorry, I forgot to type this for you. Okay, then next question, write down only the letter of the part where sound is transmitted, okay, transmitted in the form of a pressure wave. What's the answer?
It's E, the round window.
So the letter of the part where sound is transmitted in the form of a pressure wave. You said F.
Yes.
Yes, I think that is correct. Then an electrical impulse. Well, actually, I wouldn't say it's F really. It's because F is not, it's transmitting the pressure wave, but it's not, it's sort of on the way. Do you know what I'm saying?
Oh,
so I would actually say the answer for this was C, the cochlea.
Oh.
Okay, so now that differs from, because so now the inner ear is receiving this pressure wave in liquid, right? And then it's transmitting that signal, that pressure wave of sound is now going to be changed into an electrical impulse via that's question B, via what is this? What's D?
That's the auditory nerve.
Correct. Okay, so that's, yeah, it's good to make this distinction because the impulse of, well, the sound wave is transmitted in different forms within the ear. Okay, so at first it's this pressure wave in liquid. Okay, so it's in that wave form. okay, and then it's transmitted into an electrical impulse, okay, to get to the brain in order for us to interpret that sound wave. Okay, so it's good to actually make that distinction. Okay, so we said this was C. And this was D. Okay, so now a question for 3 marks. Explain the effect of the receptors in region C. Oh, again, we have this word damaged. So you need to know the function and importance so that you can be able to figure out if it's damaged, what happens. Explain the effect of the receptors in region C if the receptors in region C are damaged. Region C, what is that? Cochlea. What are the receptors? This came up in a previous question as well. What are the receptors in the cochlea? The organs of Corti. Very good. Okay, or those hair cells. Okay. So, if the organs of Corti are damaged, okay, that obviously means they cannot convert the stimulus of sound into an impulse to be transmitted to the brain via the auditory canal. Okay, so the receptors cannot convert that stimulus of sound into impulses. So if there's no impulses or fewer impulses that are going to be transmitted to the cerebrum, then that means that person cannot actually hear anything, or their hearing is impaired, or they cannot hear properly. Okay, so it comes back to the function of that receptor of that particular organ that we're referring to. Okay, so it's very important that you know functions such that you'll be able to work backwards when you're asked if things are damaged. Okay. So let's just recap quickly. The receptors called the organs of Corti, if they cannot transmit that sound impulse and change it into an electrical impulse via the auditory nerve, it means the brain is not going to get that signal of sound, which means your brain will not register that you are hearing anything. Okay, there's going to be no impulses transmitted to the cerebrum. Okay, so that person actually can't hear anything, or their hearing is impaired. Okay, for 3 marks. Okay, so now describe how the parts of the middle ear, including the membranes, assist with amplifying sound. And that's also for 3 marks. So here's the middle ear in B, okay? And we've got a membrane called the tympanic membrane. And we've got our oval window. Okay, so those are the 2 membranes that we're going to be talking about. Okay, so how does those 2 membranes, and what else is in the middle ear? These 3 ossicles, okay, that are going to be transmitting the sound. So you need to talk about these 5 components in order to answer the question, which is describe how these parts work together to amplify a sound that is received via the auditory canal. Okay, so the sound vibrations are transmitted via the auditory canal to this tympanic membrane, which will actually then vibrate. Okay, and that vibration is transmitted to the ossicles. Okay, and the ossicles are actually arranged from largest to smallest. Okay, so this malleus, incus, and stirrup are arranged largest to smallest, such that the sound is amplified. Okay, that is where this amplification comes in. Okay, so this sound is amplified by the fact that these bones are arranged largest to smallest. Okay, so the signal is going to be transmitted via these ossicles through a vibration, and that vibration is then transmitted to the oval window. Okay, and that oval window will then transmit that vibration into the inner ear. Okay. So that's all you needed to say about that, which is quite a simple 3-mark question, actually. It's not very hectic, and it's something that you should never lose 3 marks on. You should definitely be able to answer a question like this. Okay. So then, describe the role of the semicircular canals in maintaining balance. Okay, so that's these things here. And what can you notice about these semicircular canals? They're not just all in one row, they're actually in different planes. Okay, so one is coming out of the page, one is going up, one is going into the page. Okay, so it's at different, located in different planes to help maintain balance. Okay, so this is for 4 marks, this question. Describe the role of the semicircular canals in maintaining balance. Okay, so I just gave you one example of the structure of these canals, and that is related to the function, because if something is in different planes, you can tell it's actually in like 3D, so you can tell or you can tell the direction or maintain balance from being able to perceive what plane you are on, okay, in what direction you are in 3D. Okay. Okay, so that was just one thing from the top of my head, but the model answer says this: a change in speed in direction of movement, okay, is perceived or picked up by the cristae, which are found in these little, at the bottoms of these little semicircular canals. Okay, you'll find cristae. And the stimulus is then converted into an impulse, which travels via the auditory nerve to the cerebellum. Now, this needs to be mentioned because you're going to definitely get a mark for that, for knowing that the cerebellum is involved in maintaining balance. Okay, so this impulse that came from the cristae in the semicircular canals, that impulse is going to be transmitted to the cerebellum via the auditory nerve. And the cerebellum then now interprets that signal and sends an impulse to the muscles to restore that balance. So when you are on a roller coaster, for example, and you feel all dizzy, it's because that movement that your body was going through was so fast that, and those cristae were moving so quickly that the brain got so many signals of balance of your body in space and time. Okay, so your body was just moving all over in space and time that your brain received so many signals and got a little confused. That is why you feel dizzy afterwards, because your brain is busy sending signals to your body to restore that balance, to get you to stand straight and not fall over. Okay, so you have all this flood of signals coming into your muscles, and then you actually end up feeling dizzy. You carry on going in a circular motion because that roller coaster was so fast that the signals that was received by your brain from the cristae were just too fast and too many. Okay, because obviously you are on a roller coaster now. It doesn't go slowly. Okay, so think of, you could get a question like that in terms of an actual application of this. Okay. Okay, is everybody fine with that? Are you okay with this particular organ? Let's move on.
Yeah.
There's no other questions. Any questions? Okay. Okay. So now we on. I'm just getting some feedback. Okay. Okay, so here's another question now. It's got to do with the eye. And I think it's from 2019's paper. Okay, so they've given you some data. They've surveyed a number of people and checked who has blindness, who's long-sighted, short-sighted, or other. Okay, so which visual defect in the table is most common among the world population? So which has the highest percentage? Obviously, long-sightedness. Okay, in some cases where people are blind, the condition is caused by cataracts. Okay, so blindness can be caused by cataracts. Explain why people with cataracts may become blind. Define cataracts for me. Somebody.
When the cornea starts getting white spots, like milky and opaque.
Very good. Okay. So there's an actual cloudy appearance of the eye. Okay. And obviously, if something is cloudy and milky and white, it's going to actually reflect light rays and not absorb the light rays. So that light ray, those light waves are not going to come into the eye. They're going to be reflected out. Okay, so that is how that person is actually cannot see something if they have cataracts. So they are blind. Okay, then state one way in which cataracts can be treated. Unfortunately, there's no other way besides
surgery, isn't it?
Correct. Okay. The table below indicates the percentage of visually impaired people in the world suffering from different visual defects. Explain why long-sighted people need to wear glasses with a biconvex lens as a corrective measure. Okay, again, this question's coming out. They're definitely going to ask you about accommodation of the eye. This time it's for 4 marks. Okay, so it's going to be for 4 or 5 marks. Do not lose these marks by not learning this. Okay, so we've got a person with long-sightedness. How does a person with long-sightedness, how does their eyeball look? What's the shape of their eyeball? Their eyeball is it's too short. Yeah, it's not like a rugby ball look. It's more like a soccer ball look. Okay, like a squashed soccer ball. Okay, so that is a person with long-sightedness. So if their eyeball is too squashed, what it means is that the lens is less convex. Okay, it's less rounded, and that cornea is like too flat, and that eyeball is too short. Okay, so that all means that the light that is coming from any object is not going to fall directly onto the retina. It's going to fall behind the retina. Okay, because the light is going to be refracted too much by that flattened lens. Okay, so to make that lens more convex, you need to give that person glasses. Okay, so a biconvex lens will now increase the refractive power, which means it will bend the light more, such that the cone of light falls on the retina and not past the retina. Okay, not further back into the eye. So that apex of the cone of light will fall onto the retina when the light is refracted to a greater degree by the convex lens that you will give that person. Okay. So when light focuses on the retina, it gives you that clear image. Okay. So your marks are coming from the fact that you're saying the lens is less convex, or the eyeball is too short, or the cornea is flat. And this causes the light rays to fall behind the retina. A biconvex lens increases the refractive power. So refractive power is very important for you to mention in all of these things, because it means the bending of that light. Okay, you need to introduce a lens that bends the light correctly, because long-sightedness is an abnormality. Okay, it's not your eyeball. It means your eyeball is not perfectly round. Okay. And then this light will then be focused onto the retina to form a clear image. And that's where you get your 4 marks from. Okay, and then name a visual defect that is characterized by an uneven cornea or lens. This is actually me. I have an uneven eyeball, 2 uneven eyeballs.
Astigmatism.
Very good. Okay, so this is a visual defect where your eyeballs are unevenly rounded. Okay, so in some parts it's longer and some parts it's more compacted. Okay, so this unevenness actually causes images to be blurred. Okay, so it can be corrected by using a lens that is closely matched to the degree of unevenness. Okay, for people with astigmatism. Okay, and then a funny question: draw a pie chart to represent the data in the table. Okay, who remembers how to draw a pie chart? It's not something that you would usually find or that's often asked in a bio paper. You usually asked to draw a table or a graph, but this now asks you to draw a pie chart. So a recap on how to do a pie chart. This is for 6 marks actually. So how do you do a pie chart with this data here? Don't forget to carry your math set in your bio paper actually, okay? Because you would need a protractor. Okay, so to make your segments in your pie chart, you're going to say the percentage multiplied by 360. Okay, so this is 2%, 64%. Okay, so it'll be 0.64 times 360, and that'll give you the number of degrees to make that segment in your pie chart. Okay, is everybody with me on that?
Yes, ma'am.
Okay, great. Okay, now we're moving on to our endocrine system. We haven't done any of these questions yet, right? So endocrine system, here's a question from 2018, starting off easy. Identify each of the following: the gland that secretes hormone A. Okay, so in this diagram, we can see the brain, we can see the larynx, and we can see a gland X, which is found around the larynx. Please identify this gland for me. What is this gland called? That's around your larynx, your voice box.
Thyroid gland.
Very good. Okay, so the thyroid, that is the thyroid gland, and you can see now it's linked to the brain somehow. Okay, so we're talking about homeostasis and the endocrine system. Okay, so the gland that secretes hormone A. So you can see this arrow is coming from the brain. Okay, so there's a gland in the brain that secretes hormone A, which goes to the thyroid gland. And what is hormone A basically? And then you can figure out what's the gland that secretes it. Any takers?
Thyroxine.
So does the brain secrete
Thyroxine stimulating hormone.
Well, it's actually the thyroid stimulating hormone, okay? Because it's going to stimulate the thyroid to produce thyroxine. Okay, so hormone A is made by the brain and it is called thyroid stimulating hormone. So this hormone will now go to the thyroid gland and make the thyroid gland produce hormone B, which is thyroxine. Okay, so that's what we've gotten from this diagram. Okay, so the question was, the gland that secretes hormone A. So what gland in the brain secretes TSH, thyroid stimulating hormone? Do not say thyroxine stimulating hormone. It's thyroid stimulating hormone, because that is the organ on which this hormone is going to act. Okay, so it's the thyroid gland. So it's thyroid stimulating hormone. That hormone is going to stimulate the thyroid gland. Hope that's clear. Okay, so what's the gland? This will definitely come out as well. Starts with a P.
Pituitary
gland.
Very good. Okay, so the pituitary gland. Okay, or the hypophysis. Okay. What is hormone B?
Thyroxine.
Very good. Okay. Sorry, let me just type this out for you. Okay. Thyroid B, hormone B. Thyroid. Okay, then name the mechanism in the diagram that regulates the level of hormone B. So what mechanism is this showing? It's circular, which means that it is feeding back. Okay, so this is a negative feedback system. Okay. Then half of gland X was surgically removed in a person. Okay, so you know that that's the thyroid. So half of the thyroid gland was removed for a person. State 2 possible effects that this would have on the secretion of hormones referred to above. So if there's half a thyroid gland, it means it's going to be less thyroxine secreted. Okay, if the brain realizes that there's less thyroxine secreted, it means it's going to make more of thyroid stimulating hormone to make the thyroid gland make enough thyroxine. Okay, so that is that negative feedback system. If there's less thyroxine secreted, the brain will secrete more TSH to force the thyroid to make enough thyroxine. Is everybody with me? Do you understand that? Something that is something important. Okay. So that is something important to know for your homeostasis and your endocrine system. This can apply to other hormones as well. Okay. Then you are definitely going to get a question where you have to apply or read off a graph. Okay, so please be able to do that. You must be able to read off a graph. You need to know what's your independent variable, what's your dependent variable. Okay, I'll tell you all of that in the tips that at the end. Okay, but here's an example of this. Okay, so the graph below shows the effects of eating many small meals and eating fewer large meals on blood glucose and insulin concentrations in a normal person. The arrows on the graph below indicate when meals were eaten. The normal blood glucose concentration is 100 milligrams per deciliter. State what happens to the blood glucose concentration immediately after a meal is eaten. So that obviously is the same on both graphs. What happens to the glucose concentration? So we're looking at the solid line. After a meal is eaten, what happens? You can see that there is a spike in the graph. Okay, that the glucose level is increased. You with me? Everybody seeing that?
Yes.
Okay. Use the information in the graphs. Okay, this is very important. You must be able to, even if you just take 3 minutes to read the graph, okay, and understand the graph before you attempt the question, you must do that, okay? so that, because the question is asking you to get your answers from the graph, and then they're going to ask you the theory around it, or to explain the finding of the graph in light of the theory that you know from your notes. Okay, so that's very important that you are able to do such a thing. Okay, then use the information in the graphs, and now obviously you're going to be either, you're going to be asked to draw a table, graph, diagram, something. Okay, so in this case, tabulate 2 ways in which eating fewer large meals and eating many small meals affects the blood insulin levels differently. So if you're asked to draw a table, immediately you must know you're going to get a mark for actually constructing a table. Okay, so don't now do it in a point form or something. It must be in a table. So you're going to get a mark for drawing a table. By the way, where does the heading of a table go? Top or bottom? This is important. A table heading always goes on the top. So think table top. Okay, so the heading of a table must always go on the top. And you must say table 1, and what does that table show? Okay, the title must be exactly what that table shows. Okay, so in this case, it'll be showing differences between eating small and large meals on blood insulin levels. Okay, so you'll get 1 mark for drawing the table, and then it'll be 2 marks for each of the 2 ways. Okay. Let me just get you that answer. Can you see this yet? No. Okay. Okay, so let's us just discuss it quickly. We've just got about half an hour left and 1 more question. So let's motor through. Okay. So the question is asking 2 ways in which eating fewer large meals and eating many small meals affects the blood glucose levels differently. Okay, so what can you see from this graph? What is the difference between these 2 graphs? Any takers?
There are different concentrations between, how can I say this?
Between the glucose and the insulin, yes.
Yeah, I was gonna say other eating meals, but yeah, that's what I'm trying to say.
Okay, so you say you're reading it off this y-axis here. You can see that there's generally lower glucose and insulin levels. It's about 150. But here, with people eating large meals, their glucose and insulin levels are much higher. They're around 170. That's a good observation. Okay, that is definitely a point that you can put in your table. Okay.
Thank you. Thank you.
So the maximum blood insulin concentration is higher with people who are eating fewer large meals, and the maximum blood insulin concentration is lower with people who eat small meals every day. And that is actually beneficial to your health. Okay, what's anything else that you can notice from these 2 graphs? It's very important that you are able to read off graphs. Okay, so you can talk about the maximum, and you can also talk about the minimum. Okay.
Okay.
Okay, that's just 2 of them. And what else? What else does anybody else notice? You can talk about the frequency. Okay, as you can see, there's many waves here and there's few waves here. So what can you tell me about that? I'd like to hear it in your words. so that you can, you need to be able to interpret this in your own words. Doesn't have to sound great, but just put it in your own words. And we'll make it sound great. So what can you say about, see, over time, there's many spikes in insulin, but generally, if you sort of do a trend, you can see that generally over the course of the day, this person's glucose and insulin levels are maintained. Can you see that? Compare that to over this day, you can see large spikes and large decreases 3 times during that day. So this person is going to be feeling out of energy three times in that day because it's a big jumps in glucose concentrations. Can you see that? Whereas this is sort of generally maintained at a high energy level, because the minimum here, the minimum glucose insulin levels here are around, let's say 100. Okay, so if insulin levels are maintained and glucose levels are maintained at a certain level throughout the day, then it means that there's enough blood, there's enough sugar in the person's blood. So they're not going to feel drained so badly. They're not going to feel tired and out of energy, okay? Because there's a constant high level of insulin in the person's, low level of insulin in the person's blood. So there's going to be enough sugar in that person's blood to give them energy throughout the day. Whereas this graph shows that there's actually much lower levels of insulin. It drops. So you hit a high and then you drop your energy levels, because look at this, it's only about 80, okay? And that happens in a very conspicuous way or a very stark way 3 times in that day. So after breakfast, after lunch, after supper, this person is feeling drained and tired because their insulin levels are very high after these big meals. And then a few hours later, it's very low. So they're feeling, so when it's very low, that means they're either feeling very energetic and very tired. Okay, whereas this person's levels are more, I wouldn't say constant, but it's not such a big gap between these ups and downs, if you're understanding what I'm saying. Okay, so just reading off that graph, you can put it into your own words to get 2 differences. Okay, so you can say large changes in insulin concentration versus small changes in insulin concentration between fewer large meals and smaller frequent meals, respectively. Okay. Also, what will help you make sure you get those marks is if you quote figures. So say at 8 o'clock, there's 170 milligrams per deciliter. Okay, so quote from the graph in your answer. That will show the marker that you are able to read off this graph. That's very important. Okay, don't leave those out. So be very specific. Okay, so explain why eating small meals per day is better for a diabetic person than eating fewer large meals a day. First of all, you need to know what is a diabetic person in this question. This is for 4 marks. Okay, so what is a diabetic person? It is somebody whose blood glucose levels are irregular. It's not controlled properly by the release of insulin and glucagon. Okay, because the pancreas is not functioning correctly. Okay, so it's very important to define that first. Okay, so a diabetic may not produce sufficient insulin, okay, which means that that person is going to have a very high blood glucose level. Okay, so in order to, some diabetics have to take insulin in order to decrease that blood glucose level. Okay, so when eating smaller meals, there's less fluctuation in that insulin level, and there's less of a maximum insulin level and less of a minimum insulin level. Okay, compared between those two graphs. So when many small meals are eaten, there's less glucose entering the body at a given time. Okay, so less glucose enters the blood, which means that less insulin is needed at the time. So that is why diabetic people are advised to eat very healthy meals, because they do not want these large spikes in glucose levels in their blood. Okay, so when eating smaller meals, there'll be less glucose because you're eating a healthy meal and it's smaller. So there's less glucose in the blood, which means that less insulin is needed, okay, to correct your levels. Your levels should be about 3.5 to 5.5 millimoles per liter in the blood. Okay, so less insulin is needed if you're eating these small healthy meals. And this will allow the blood glucose level to be more in the normal range. Okay, so if the person's blood sugar levels are maybe 5 or 6, that's okay. That's within that normal range. Okay, so this is a very good application question that is likely that you are likely to get something similar to. Okay, it might have been a little bit tricky and you might have found this a little bit difficult, but I suggest you practice questions like this from past papers if you are uncomfortable with this question. Okay, it was for 10 marks, so you don't want to get all of those incorrect. Okay, and to end off, we also have another question on diabetes. So let's just try and do this a bit quickly. Okay, so name the gland that secretes glucagon. Okay, sorry, so you have to read all of this in your paper. Take 2 minutes, take 3 minutes, go with a highlighter and highlight your key points in this little blurb that they've given you. Okay, so make sure you go with a highlighter. Okay, they've given you an investigation that was conducted on 100 patients with type 1 diabetes, and they were also glucagon deficient, okay? And they were given the same amount of food and water for 3 days, and then they were given adrenaline, okay? They were all given adrenaline, and then the blood glucose concentration in each person was measured after they were given adrenaline, okay? And then obviously the response was seen. So we're looking for the response of adrenaline in these patients with type 1 diabetes. Okay, that was just a quick summary of what that said. Name the gland that secretes glucagon. The gland. Quickly, let's get some answers.
Pancreas. Pancreas.
Is the pancreas a gland?
Yes. Yes.
It's a sort of a tricky question. It is an organ, but the answer that they're looking for is actually the islets of Langerhans within the pancreas. You see, so you got to be very specific. You might get half a mark for saying pancreas, but we're trying to get that full 1 mark. So you can say pancreas, specifically the islets of Langerhans. You with me?
Yes.
What if you say alpha cells?
That's not a gland.
Oh.
It's cells. Yeah. Okay, so you got to be specific and say pancreas, islets of Langerhans. Okay, so identify the independent variable in the investigation? What's the independent variable? What is being administered? The thing that we are controlling in the investigation. We are controlling the adrenaline that's being administered to these patients. Okay, so adrenaline is the independent variable. Then state 3 other factors that should have been kept constant during the investigation. Okay, there's many. I've just given you a few. Just from reading this, we can say, okay, things that need to be kept constant are always factors that can affect the reliability and validity of an experiment. Okay, so the type of food that the patients were given to eat, the concentration of adrenaline given to the patients, the amount of adrenaline given to the patients needs to all be the same. Things that you need to keep the same such that they do not have a negative impact on your reliability and validity of your experiment and the data that you're going to receive from doing the experiment. Okay, so you're trying to get reliable data, so you can't have confounding factors that are going to affect your experiment. You're definitely going to get a question like this where you are asked about an experiment. Okay, so please, as I wrote it in my tips, you must know how to interpret an experiment that's been done. Okay. Okay, so the other things you would keep constant or you keep the same would be the measuring tools, the person measuring the glucose concentration. Okay, all like human error factors. You're trying to eliminate human error as well. Okay, in this experiment. So those are types of things you would keep constant. Okay, explain why the blood glucose levels were measured before injecting adrenaline on the 3rd day. Okay, so they were given food and water for 3 days, then the blood glucose levels were measured, and then they were given adrenaline. So why was the blood glucose measured before adrenaline administration? Remember, what are we trying to investigate? What are we investigating? We're investigating the effect of adrenaline. So you need to know what is the baseline. What is this person's body doing before adrenaline is administered? So then when adrenaline is administered, you have something to compare to. Okay, so this is the baseline. You're trying to get a baseline blood glucose level before the administration of adrenaline, before you can see the effect of adrenaline and be able to compare blood glucose levels before and after, because adrenaline is definitely going to affect the person's body. Now, why was adrenaline injected instead of given orally, which comes to the point of what is adrenaline? Anybody know? What is adrenaline? It is a hormone. And what is a hormone? It is a protein. Okay, so proteins, if you're going to eat a protein, it's going to go into your stomach and be broken down. Okay, so you wouldn't want to take adrenaline orally because it's just going to go into your stomach. If you take it intravenously or it's injected directly into your bloodstream, it's not going to be broken down, and that adrenaline can now go to the exact point that it needs to act on. Okay, it's a hormone. It's going to act on a distant thing, a distant organ. Okay, so it can now directly travel through the bloodstream to the organ where it's going to cause an effect. Okay, so you wouldn't want to take that orally because it's just going to be broken down, because adrenaline is a hormone, which is a protein. Okay, that was for 2 marks. Then explain what would be the expected results after adrenaline was injected into the patients. Now, what do you think is the function or the effect of adrenaline in any patient?
It prepares the body for emergencies.
Correct. So how does it prepare the body for emergency?
increase heart rates,
Yes.
blood pressure.
Yeah, correct.
Breathing.
Most importantly, adrenaline increases blood glucose levels. Because remember now, if you are in a flight or fight response state, your muscles need energy. They need to be able to fight or run away. So your muscles need that boost of glucose. Okay, so that is what adrenaline does. It rapidly increases the glucose levels in the blood. So that is what we expect to happen here after we've administered adrenaline to these patients. Okay, we expect that their blood glucose levels will increase. Okay, then give a reason for the use of 100 patients in the investigation instead of only 10. This is a scientific method question. Why would we use more samples? To increase the reliability of our experiment. You are definitely going to get a question, maybe even in multiple choice or something, about reliability or validity of an experiment. Okay, and you must be able to know that you have to have a large sample size in order for your results to be reliable. Is everybody with me? Are you okay with this? If you are uncomfortable with this type of question for 12 marks, please do some practice on these. These are application questions. Okay, so they are a bit more difficult. They require you to be able to interpret an experiment or read off a graph. That's interpretation skills. So you need to be able to read off graphs and interpret these investigations. Okay, very important. And I'm just going to end off with some key study tips for these sections. Okay, particularly the responses to the environment, human responses to the environment, and endocrine and homeostasis. Structure and function is key. Okay, so you must know the structure of the brain, the neurons, the spinal cord, the eye, the ear. You must know the structure, okay? Because you have to be able to label these things. And as you see from all the questions that we've done, they're always asking you the function, but they can also ask you the structure of something in relation to its function. For example, the structure of the eye in relation to how it is functioning. For example, if the eye is more rounded, then it's going to function differently. It means you're going to be long-sighted. Okay, so structure and function is absolutely important. You must know that for these two sections. Okay, and then as I said, the scientific method for application questions, you must revise that. I mean, you've probably done it I don't think early in the year, but maybe in previous years, but please revise what is an independent variable, what is dependent variable, controlled variables, why do you need a control, how do you increase the validity or reliability of the experiment, and that links to how you're going to draw a graph. You need to know that your independent variables go on your x-axis and your dependent variable goes on your y-axis. You need to be able to draw that graph from an experiment, for example. Okay, they could even ask you the hypothesis of an experiment. Okay, so that is a statement of difference or no difference between your two variables, your independent and dependent variables. You're saying something has an effect on something else. Okay, then you need to make summary tables. For example, in this endocrine section, you can make a table which says the gland, the hormone that gland releases, and the function of that hormone. Okay, so you can make a whole table about that. For example, the thyroid gland makes thyroxine, which regulates metabolic function. Okay, so make a nice table to summarize these things for yourself. Okay, it's also in the study guide, but I'm sure you'll even be able to download this somewhere. of the net. Okay, it helps you to study and to be able to quickly answer these types of questions. Okay, you must be conscious of your time management. Don't waste time on these one-word answers that you can just learn off and get that 1 mark in less than 1 minute. Okay. Then you need to also know about the diseases and disorders. For example, in the endocrine system, we needed to know about diabetes, thyroid disorders like hyperthyroidism, hearing defects like deafness or middle ear. What does a middle ear infection do? For the brain, maybe how does drugs affect the brain? Okay, or diseases of the nervous system like Alzheimer's disease or multiple sclerosis. Okay, so just read through that and be aware of the diseases and disorders that's in these, that could be asked about in these sections. Okay, so I hope this whole revision session was informative for you. I hope you got something out of it and that you were able to understand the extent to which you need to study more, maybe, or if you manage with all those questions. Okay, the best thing you can do is go over all these past year papers, and whatever you don't know, revise those sections. Okay, so I'm just going to now transfer you to any of our or refer you to our chat. Okay, you can see that this video will be posted. Okay, the link is there in the chat. Okay, and if you would like to refer to any of the resources that we have for you, you can go to the College of Agriculture, Engineering and Science. Okay, and if you can also please fill in the register at the end of this section. The link is in the chat. Okay, it's posted by Naomika. It's forms.office.com. Okay, so if you can fill in that register quickly, that would be great. And that would be, if you want to also listen to this recording again, it's going to be posted on our teaching and learning site, which is located on the UKZN website in the College of Agriculture, Engineering and Science section. Okay, so I hope you all got something out of this and learned something. It was a good 2 hours of revision, which you have under your belt for your life science paper, and I wish you all the best. Please come back on Thursday where we will be doing the rest of the sections for paper 1, okay, which also has a big section out of 41 marks, 13 marks, and 8 marks. Okay, so please come back on Thursday. Okay, and we're going to play out with a video on how you can apply to UKZN. Okay, thank you everybody for your time. Take care.
Experience is the opportunity to be part of a community and to network with not only our world-renowned academics, game changers and leaders. They do say that the extent of one's success can be attributed to those they rub shoulders with. Therefore, you must surround yourself with like-minded people. So, how do you become part of a community that is committed to your self-development? How do you become part of UKZN? First, you have to apply.
All applications for undergraduate studies at UKZN are processed through the Central Applications Office, CAO. This is for both your course and student residence application. The application process is as follows. Step 1. Consult the CAO handbook. This handbook provides a list of all qualifications available at UKZN. Step 2. Have a look through the various courses offered and choose a program that interests you and for which you meet the minimum requirements. As one of the top 5 universities in the country, UKZN promotes academic excellence. so meeting the minimum requirements does not guarantee you a place to study. Step 3. Find the program code linked to your chosen qualification. This is the code you need to enter in your application form. Okay, let me show you something. You see, a code has 3 parts. Example, for BCom in accounting, the code is KN-P-BCN or KN-W-BCN. Crazy code, right? Well, basically, the KN stands for UKZN, the middle part stands for the campus of your choice, P for Pietermaritzburg, W for Westville. Then BCN stands for the actual course. Make sure to get these codes right. Step 4. Use this program code when completing your CAO form, which is available from the CAO website. Your application will be sent to UKZN. You can always check the status of your application using your CAO number, ID number, or passport number on this website, www.caocheck.ukzn.ac.za. For more information, visit www.cao.ac.za.
It's that easy. Pay close attention to closing dates for qualifications. For example, MBCHB closes on the 30th of June, whilst other undergraduate programs close on the 30th of September. What are you waiting for? Apply now and shape your future today.
If there are any grade 11 students, you may send your questions to Life Sciences. Sorry, let me just get the correct email address. You may send it to Life Science Helpline at ukzn.ac.za. I apologize that we didn't get time to go through any grade 11 content, but I do see some questions on the chat, and you're more than welcome to email specific questions to us, and we will respond to you within 24 hours. I see that you're writing on Monday. Are there any grade 11s here currently? I don't mind just getting a quick show of hands. But if not, it's fine. We will go through that. Oh, okay. We have a few. Okay. So I saw a question about the nephron. Okay. And filtration. Is that something that you really need some revision on? I can do a crash course in 5 minutes. I also recommend that you watch YouTube videos. Those are surprisingly helpful. I would recommend Khan Academy or short little videos where you can just revise. And it also helps to see how an organ works in an animation. Okay, so I would recommend that you do that. Let me find my notes on the nephron for you. Shame, at least you've got a head start for Matric. You did some revision for Matric already. Okay. Okay, so what you would need to know about the nephron, definitely structure, you need to be able to label it, and what each part of the nephron does. Okay, I don't actually have a slide for you, but I'm just going to explain it. Okay, so I'm not sure if any of you have the study guide. Okay, but definitely the nephron in terms of labels and function, it is going to come out. Okay, so you need to be able to explain how a nephron functions if the salt levels are too high, or the water, your body's water content is too high. Okay, you need to be able to know how the nephron is going to work to rectify those levels. Okay, so the nephron has, I'm sure you've seen in the chat from my colleague, Dr. Namisha Singh, she's written here that nephrons have a 3-step system of filtration, which is reabsorption, filtration, reabsorption, and secretion to form your urine. Okay, so those are the cells of the kidney that are actually going to make your urine. Okay, and by filtering out every liquid in your body, it is going to maintain your sodium levels. Okay, and it's going to adjust your water content and your iron levels as well. Okay, so the nephron consists of 2 portions. That would be your, anybody know? Your renal corpuscle and your renal tubule. Okay, now what is the function of the renal tubule? It's going to actually filter your blood, okay? Or filter the liquid portion of your blood, okay? So your renal corpuscle consists of 2 parts, the glomerulus and the Bowman's capsule. Okay, that's important. You need to know the structure of those 2 parts, okay? And then you have your renal tubule and the ducts and the blood supply to that nephron, okay? So importantly, you'll probably be asked on glomerular ultrafiltration, okay? And that is where the blood in the glomerulus is under a certain pressure and now has to be passed through that nephron, okay? And when it does that, that pressure forces useful and harmful substances through the glomerulus and the inner wall of the Bowman's capsule. Okay, so I have a summary here for you, but I'm just going to read it to you so that you can maybe jot it down as we go, for those of you who would like to do that. Okay, so as I said, that pressure of the blood flowing through those cells is going to also filter out the harmful and useful substances and ions. Okay, the tiny pores in the capillary wall of the glomerulus has specialized cells called podocytes, and those form the cup of the Bowman's capsule in a cup shape. Okay, and they actually filter out everything that is in the blood. Okay, and it's selectively, very important that it selectively allows certain substances to pass through. Okay, so substances that will definitely pass through will be definitely water, glucose, amino acids, urea, and uric acid. Okay, the fluid, which is with dissolved substances inside it, enters that capsular space and is called the glomerular filtrate. Thank you very much for this diagram. Okay, so you can see the Bowman's capsule there on the right-hand side of that diagram. Okay. Then the glomerular filtrate. So once this blood is now entered the nephron and it's going through this thing, it's referred to as the glomerular filtrate. Okay, it passes along the renal tubule thereafter, after going through this Bowman's capsule. Okay, so now it's entering the stage of tubular reabsorption. So that is in that long part. So in the middle of the diagram, this long windy part is your your renal tubules, where tubular reabsorption is going to occur. And this is where 99% of the water is going to be reabsorbed into your body. Okay, so each part of this tubule has a different function. Okay, the proximal convoluted tubule now allows water and useful substances like glucose to be selectively reabsorbed, which is a very important job of the nephron. It needs to reabsorb that water and glucose, otherwise you're going to just be constantly weak and needing to use the toilet. Okay, so the process of selective reabsorption also occurs in this section, but that is done in certain specialized cuboidal cells that you'll find in the renal tubules there. Okay, so this filtrate will now pass through the convoluted tubule and enter the very important section which you need to know the function of, the loop of Henle, which is down there. Oh, I can't see it on this diagram, but you need to know the loop of Henle. Okay, the function of the loop of Henle is to ensure that the medulla has a high concentration of sodium ions. Okay, sodium is extremely important in your body. You will never be able to conduct any nerve impulses or make your muscles do anything if you don't have sodium. Okay, so sodium allows your muscles to work. So reabsorption of sodium from the blood is extremely important, and that is facilitated by the loop of Henle. Okay, so sodium ions will diffuse passively into the tubular fluid along the descending limb of the loop of Henle. Okay, and then lastly, you have your tubular excretion. Okay, so tubular excretion now occurs when you have substances that you actually want to get rid of. Okay, so tubular excretion is the movement of substances from the blood into the tubular fluid that you want to be excreted. Okay, so excreted substances like hydrogen ions, potassium, creatine, certain drugs like penicillin, okay, those will be excreted in this part of the functioning of the nephron. Okay, so the pH of the blood is regulated by this process. Okay, so it gets rid of too much hydrogen ions, or it keeps some hydrogen ions. It will affect the pH of the blood. Okay, and what else can I say about tubular excretion? It's the final step that actually processes what your body will get rid of in the urine. Okay, not all of the detail. For example, if you look at the diagram that you see there, I don't think they're going to ask you about sodium and potassium pumps and the energy required, but they will definitely ask you what is the details of tubular excretion in terms of the nephron. So in which part of the nephron it occurs, how it occurs, what would be excreted. Okay, so those types of things that you might need to know. Okay, so, and then once your body is ready to excrete a certain amount of solutes and water, that is when the urine is ready for it to be excreted, and it will pass through the bladder and you will excrete it. There's a nice summary for you in terms of urine formation. Okay, this is a very nice summary. Okay, for you to just quickly go through. You can see that things are reabsorbed and back into the blood in B. Okay, and certain fluids are reabsorbed into the nephron at C. Okay, so you've got your 3 big steps: glomerular filtration, tubular reabsorption, and tubular secretion. Okay. My colleague, Dr. Nimisha Singh, would you like to add anything? Because I've just done this in a rush quickly for the grade 11s, but would you like to add any details there? Remember, they can also ask you about the role of the antidiuretic hormone ADH in osmoregulation. That also may come out in a question. So please also know that. What happens when there's a high levels of ADH in the blood? So will you get concentrated urine or dilute urine? Okay, so it's very important for you to know those types of things. Over to you, Nimisha.
Hi, everyone. Can you hear me?
Yes, we can.
Okay. All right. I had some mic issues. So what Mrs. Jerusha has been saying is quite correct in terms of the urine formation. I did send a link on the chat for past year papers with the memos, so you can see the type of questions they ask, but it's basically what Jerusha has been saying in terms of answering the question. So you really need to just know the actual steps, the 3 steps that you see here, and what ions are involved for the specific functions in terms of salt level regulation. Because remember, our primary dietary salt is sodium chloride, and sodium chloride is important for various pump mechanisms within our cells that allows for reabsorption of water. So you just need to know the actual process that happens within the glomerulus of the ion exchange that allows for the formation of creatine and the other metabolic waste secretions that will eventually get to your bladder for waste disposal. So the link that I did send, it has a nice breakdown of the different types. So they may ask you an image of the actual entire glomerulus and ask you to label. I was trying to share one of the papers, but my tablet is refusing to be sharing the PDF. So from the link I sent you, it'll be very helpful with that one. So basically, they'll give you probably a structure of the actual nephron like this, or they'll give you the whole urinary system that starts from the kidneys going down to your bladder, and they'll just ask you about the specific organs, what are their functions as well. As well as you'll see normal MCQ questions. You just need to know basic functions about that. Otherwise, the crash course explanation was perfect for it. Any questions?
Yes, regarding the podocytes, I still don't understand what role does it play. I really do not get it.
Sorry, could you repeat the first part? It just broke up for me a bit there.
The podocytes, I still don't understand what role does it play. I don't get it.
The podocytes?
Yes, the podocytes.
Okay. Hold on for me, if I can get it.
Okay, so podocytes are they are specialized cells, okay, that are found in the wall of the glomerulus. Okay, so I'm just trying to, okay, there, if you look on the diagram, you can see where the Bowman's capsule is, like you can see that that cup shape, right?
Yes.
You can see the blood vessels that's going into that cup shape. Now, along that cup shape, you're going to have these specialized cells, okay?
Yes.
Right along that cup shape on the inside there. So the blood is coming into contact with these podocytes, okay? It forms the wall of that inner cup of the Bowman's capsule, okay? So those podocytes now they have these slit pores which allow certain substances to pass through. So basically they are like these specialized cells that will only allow certain things to enter the nephron and certain things won't, by these little slits, these pores that they have in them. Okay, so they're just specialized cells that will allow certain substances through from the blood into the Bowman's capsule or not.
Okay, thank you.
Okay, here's a closer look at them.
The podocytes are the green towards your bottom right hand corner.
Yeah, so those cells are going to be what actually allows comes into sort of direct contact with the blood vessels and allows certain things from the blood to come into the Bowman's capsule or not. Okay.
Okay, thank you very much.
You're welcome. Any other questions?
No further questions.
Okay, great. All the best for Monday.
Good luck, everyone.
Thank you.
Just remember to read
Thank you very
questions.
much.
Yeah, you can, if you have any questions at any point this weekend even, you can email them to lifesciencehelpline@ukzn.ac.za and we'll get back to you as soon as possible before Monday. Okay, I think we're done.
Yeah, everyone has
left.
Yes, everyone's left. Yeah.
Great session, Jerry.